All right, so we've covered how to transfer DNA from one cell to another, either through conjugation or through transformation. But you can also transfer DNA between cells using phages. So bacteriaphages, so these are viruses that have bacteria as their host, and they reproduce via bacterial genetic recombination called transduction. So here we can see what a phage looks like, and so they look kind of crazily like you might imagine a phage to look like, where they have these arms, these kind of like spider-like arms, and these spider-like arms are used to actually attach to the host cell. So this is part of the way that they're able to recognize the bacterial cell that they're trying to infect. And so you can see this cool EM pictures of some of these phages. And so when the DNA and protein, so they can have both DNA and proteins within the head of the phage, you can see that reflected in this brightness within the head. And then as these phage infect the bacterial cell by injecting the DNA, you can see that this part of the region actually empties out, and you can actually kind of even see like pieces of the DNA actually getting injected into the bacterial cell. So this is a T4 phage that infects E. coli. So it has the head, the neck and collar region, the core region, the sheath, and then the end plate. And so these are the the fibers are kind of like the legs. And so these are the regions that must be assembled together in order to create a virion particle. So one of these is a virion. And so, this DNA is able to infect the cell, it takes over the host machinery, and then that instructs the cell in order to create a large number of these head, neck and collar, core, sheath, and end plate proteins using this DNA as the blueprint, and then it's able to assemble a large number of new virion particles that are able to exit the cell once the cell lyses in order to infect other bacterial cells. So normally what happens is the phage finds a host cell, it's absorbed to the bacterial host cell, the phage DNA is injected, the host DNA is degraded, so this might contain proteins that go in there and actually chop up the DNA into pieces, and then the machinery is taken over in order to replicate the phage DNA, so you make multiple copies of the phage DNA using the DNA polymerase, usually a phage will encode its own polymerase in order to replicate that. And then you create a large number of these phage DNA genomes, you create the proteins that are necessary for assembling the phage particle, and then this is all assembled together. And so part of this assembly requires actually taking the phage DNA and assembling it properly into the head part of the virion particle. And then once this happens, the host cell is lysed and phages are released. So normally this just kills the cell, and there's no sorts of DNA transfer that occurs. But it was shown that actually you can use recombination between bacterial cells using bacteriaphages. And so this was a very famous experiment from Lederberg-Zinder, and they used Salmonella, which is an auxotrophic strain, and this led to the discovery of phage transduction in bacterium. And the idea is that you have two strains, so LA2 and LA22, and they have opposite genotypes at the following 4 genes. And so this allows you to distinguish and determine when recombination occurs. And these are these are separated by a membrane. So this membrane allows the media to pass back and forth, but the cells are not. So the filter size of this membrane is smaller than the bacterial cell size. And then you use pressure to basically push liquid back and forth. And so they did this, and they incubated on minimal media that required that only a prototroph could grow on. So this strain could not grow on it, and this strain could normally not grow on it because it was minimal media, and so the strain has to be able to produce phenylalanine, tryptamine, methionine, and histidine, and the two strains lack two of the genes. But they found they could actually get recombinants that could recombine out these two alleles with these two alleles. And that was possible because these had a phage within it, and this phage was actually able to infect the cells and then transfer across this membrane to the other side. And so how does that work? So this works because phage production is not perfect. So sometimes instead of packaging back the phage DNA into the phage particles, bacterial DNA is incorrectly packaged. So again, we see this life cycle of the phage. Phage DNA is injected, and the DNA is broken up into pieces. We replicate the phage DNA, we produce all of these proteins, viral proteins that assemble, and then the full phage particles are assembled together. But what you'll notice is what can happen with a rare amount of times is that instead of packaging up the phage DNA, we've packaged up one of these pieces of bacterial chromosome DNA. So this cell lyses and this viral particle is released in the environment. So even though it doesn't have the viral DNA, it's still functional and able to do its thing. And so once it finds this host, it's able to inject the piece of DNA into the cell. And so instead of having the DNA that will encode the DNase that will break up the DNA, instead what you have is this bacterial piece of DNA, this linear piece of DNA that could then be potentially used as a site of recombination. So again, if there's homology with this piece of DNA, this linear piece of DNA in the circular host chromosome, you can have recombination occur, crossover occur, and so then you can replace part of the genome with the other genome. So again, recombination is used, so recombination between a linear piece of DNA and the circular chromosome is necessary for this process to lead to any sort of bacterial genetic transfer, but it's possible as long as there's homology. And so transduction is the way that this phage particle is able to actually transfer DNA, bacterial DNA, between cells.